Hello everyone, welcome to today's video, and in this video I will be discussing IgG antibody and IgG antibody subtypes. On the screen you can see all the subtypes are there. We have IgG1 right over here, then you have IgG2, and then IgG3, IgG4. Please excuse me, my voice is not clear. I had the infection and then I suffered for quite a few days. Now because of that I have a heavy voice. So it's not similar to the routine voice that I already have. Anyways, so the problem is the infection. So it is very, very relevant for me to make this video on antibodies. How antibodies, they are important in protecting our body from infection, and when there is a breakage of the protective barrier, and then infectious agents, they can take over and they can cause diseases in our body. And when these the barrier is being breached by the infectious agent, only molecules that can save us will include the molecules of adaptive immunity, and one of the important molecule right here is the antibody, and which is IgG and IgG antibodies. They will have further subtypes: IgG1, IgG2, IgG3, and IgG4. There is a subtle structural differences that you are seeing, especially in case of the molecular weight and the disulfide bonding in all these antibodies, and we'll also discuss that in detail, right? So we'll start with the antibody structure first, and in the structure what you're seeing is antibodies. Basically the size of antibody is 150 kilodalton. So kilodalton is the unit to measure the molecular weight. So this is, it seems like I've written ISO. So it's 150 kilodalton. That is the molecular weight of this entire molecule. I'm talking about the entire antibody molecule, right? And then in that you have a typical molecular structure which is tetrameric in structure. Tetrameric means tetrameric. So let me write it out. Tetrameric structure means it will have 4 different chains. So now you can see it will have chain number 1, which is this, and then this is the second chain, and these are in green color, which are also known as heavy chains, and this is also the second heavy chain, and then you'll have this light chain over here, and then second light chain over here, which are in the yellow color. So this is how it is tetrameric in nature. That is why it has the word called tetrameric, because it is basically made up of 4 chains, right? And next, if we move further, IgG antibody basically will have 150 kilodalton. That is the molecular weight I mentioned, and it's the tetrameric entire structure, right? So that is what I've already explained. Now moving further, in case of the antibody, you have 2 heavy chains, and that will be indicated by letter H, and various domains will be there that we'll talk about later on, and then 2 light chains, right? So I hope now it is clear that these are the regions of the light chain. And now because of that, you have this particular head of this Y-shaped structure. So this head I'm talking about, right? So this is the region where the variable region of the antibody is, and where it can bind to antigens. So antigens are the molecules that are the foreign molecules, foreign protein molecules, peptides. They can interact with the components of the immune system. Now let's move further. These 2 chains, now we need to understand what are their molecular weights. So these 2 chains approximately will have 50 kilodalton. We are talking about heavy chain, that's why it is known as the heavy chain. And in case of light chain, it will have around 25 kilodalton molecular weight. So we are talking about KD. This is how you write KDA, okay? So the kilodalton, and this will constitute the light chain, as I already told you. So it will not have that region, right? That region, which is extra in case of heavy chains. So approximately 25 kilodalton is missing in this case. So I hope it's easy to remember in this sense. So if you remember, this is the 50 kilodalton, and then minus 50, 25, that is the light chain you have. Okay, moving further, you'll have disulfide bonding right over here. Let me create these disulfide bonds right over here. So this is the disulfide bond which is keeping not only the heavy chains together. So this is important not only for heavy chains, but it's also important for heavy and light chain interaction. This is how this entire molecule will remain intact like this. So it's a globular molecule. So this is how it's going to intact. It's going to be intact, and it can basically be stable inside the body. So disulfide bondings, it is very, very important, and we all know when we talk about disulfide bonds, cysteine residues are important in case of the antibody structure. They will form the disulfide bonding together. Okay, let me take a little bit break over here and talk about the infection process. Infection process happens when there is foreign viral particle that enters inside the body. And in my case, if I take the example of me, what happened to me is basically I got throat infection. Right, so I got the throat infection, and then whether it was viral, whether it was bacterial, that still is very, very difficult problem, right? Whenever there is infection or cold, I would say, right? So cold is viral, we all know that. And then you can also have bacterial infection in many cases, right? It will be by the bacteria. So in my case, I had the cold because it was very acute, runny nose was there, and then I had fever. After that, my body recovered. So I haven't took any kind of antibiotics, and I also recommend others not to take antibiotics unless a doctor is prescribing you antibiotic, and my body basically recovered. It took time, but slowly I was able to recover from the cold. So it's very important that you go to doctor, and then if doctor tells you take the antibiotics, then only you take the antibiotics, because that is the advice you are getting, because maybe the infection is serious. But usually when you have mild cold, then you should not take antibiotics. What will happen? Any kind of virus will enter inside the body. It will interact with immune system, and it will generate memory cells, right? I'm not going into the detail of how it will happen, and it will generate antibodies. Then they further will bind to virus, and they will neutralize it, and then you will have that immune memory in your body, so that you are protected from subsequent infection. That will not happen in this case, especially if it is bacterial. But in case of a virus which usually don't have any kind of antiviral therapy available right now, I think there should be either a vaccine against cold. And if we have vaccine against cold, that would be nice. In other countries like USA, you have shots. They're called flu shots. And before there is a seasonal change, you should get the flu shot, and you will be recovered from that, because it will work as a kind of immune, providing you the immunity against the infections. So in India, I think we also should have something like this. Anyways, discussion further. And in many countries and many developing countries, they don't have flu shots, and I think that flu shot should be made available to all of them, right? So okay, I've made this slide really busy. So let's move on to the next one. Now we all talk about this Y-shaped structure of antibody, and I'll tell you why it is Y-shaped. You know that why it is Y-shaped. Actually, it is not like this. It will have the globular structure like this. It will be, how the protein molecules they appear. So it will be very, very unique in structure. I don't know. So this is how it's going to be. So it's going to have globular structure, but typically it will acquire this typical Y-shaped structure. So Y-shaped structure, and why it is Y-shaped, what is the significance of that? One significance is that you have antigen binding site over here, and then it can bind to Fc receptor in the cell. So this is the Fc region of the antibody, and this is the Fab region of the antibody. Right, AP antigen binding happens over here. This is the crystallizable part, so that is why it's known as Fc fragment. Next, moving on to various domains. So in case of heavy chain, so HEAV, heavy chains, you have C constant region. Now, as I already told you, the antibody will have region 1, region 2, region 3, and all these regions are constant regions, means they will not have that much variability, but the top one, the region which is right over here, will have variability, hence known as variable region, because it will bind with that antigen, and antigen can vary, right? That is the property of the antigen, it can vary. So all those other regions, they are known as the CH1, constant region of heavy chain 1, 2, 3, and then you will have further you will have hinge region. Hinge region is very, very important. This region is called as the hinge region. Why? This region is important. Why it is important? Because it is important for the flexibility of the antibody molecule, because of that it can move in various directions, right? And hence, because of that, it has the ability to basically bind with the antigen effectively, and also be stable under various conditions. So that is the importance of a hinge region. Now moving to the VH region, as I already told you, V means variable, and H means the variable region of heavy chain, right? If the variable region of light chain will be written, it should be written like this: variable of light chain. Okay. Now moving further, we have various antibodies. So now let's start with IgG1. Now it is very, very important for us to understand a few things. There are different subtypes of the antibody, right? There are various subtypes. You have IgG1, IgG2, IgG3, and when it comes to IgG1, it's a subclass which represents highest 60 to 65% of the IgG antibodies. They are these antibodies, IgG1 antibodies. They are primarily responsible for basically the immune response, which we call immune response, right? Immune response, they are the one who are responsible for neutralizing virus, bacteria, and all other immune functions. IgG1 are the antibodies. So this is the structure. You can see it's a typical structure. Whenever you see antibody structure, that will be the structure of IgG1 antibody. Okay, so 60 to 65% of the antibody. And then moving on to IgG2. Now, what is the percentage of these antibodies? Approximately, I'm going to say 25%. So 20 to 25% is the IgG. And it's responsible to antigens which are found on, basically antigens which are polysaccharides, right? Polysaccharides and carbohydrates. Usually those are the antigens that are associated with IgG2 antibodies. Okay, and now moving on to the next part, IgG2 antibodies, another important point that I want to write is, especially in case of young children's, respiratory infection. If there are respiratory infections, in case of young children's, it is mostly IgG2 antibody is involved against repeated respiratory infection. So deficiency in IgG2 will lead to the respiratory infection. So I hope you understood, right? If there is any deficiency in IgG2, there will be the respiratory infections in case of children's. So that is something that has been observed. Now let's move on to the IgG3. Okay, IgG3 is, now again the percentage is going to go down. So 5 to 10% of the antibodies, they are total IgG3 antibodies. Now look at the difference. Difference is basically in the number of disulfide bonds are high in this case, right? And it's significant antibodies against proteins or peptide antigens, and it's not that prevalent. Okay, these type of antibodies, they are known to produce against proteins or peptide antigens, like similar to IgG1, but as compared to IgG1, this antibody, IgG3 antibody, is not that prevalent. And research is going on in this area to identify why this is the scenario. Now moving on to IgG4, this is the antibody, which is again, now it's going to be, it's less than 4%. This is how it's going to come up to the 100%, right? Less than 4% is the IgG4 antibody in our body. Now, this is less abundant or least abundant, you can say, and it's not going to react with polysaccharide antigens. This is one of the factor, and it's also responsive against the protein antigens, which is basically in most of the cases, or polypeptide. And more these isotypes or more these subtypes of these antibodies are being looked into, and why there is increase of the expression of these type of antibodies in specific conditions. A lot of research is going on into that one, why our body is producing different types of antibodies. Like, okay, coming on to the final slide and look at this. So there is slight difference in the disulfide bonding right over here and right over here, and there is also this conformational change you observe in IgG2 antibody as compared to the other antibodies. And obviously its molecular weight a little bit, it's going to be higher, on the higher side, right? Okay, let's talk about some of the important functions of the IgG antibodies, which includes number one, infection control. So infection, as we already discussed, that is the important function of IgG antibodies. It's going to control the infection. Second one, it is the only antibody that crosses placenta. So the baby that is still in the body of the female, it's going to provide immunity to that baby. So neonatal protection. It safeguards the newborn early on, right? So that is important. Opsonization of phagocytosis. Should I write it over here? Okay. Opsonization means covering up the foreign infectious agents. They can attach to the infectious agents, and because of that, they can be phagocytized by the immune cells very, very easily. So opsonization is basically covering the infectious agent, and then phagocytosis, right? So that is the another point. Now let's move on to another one, immunization. In case of vaccines, the responsive functional molecule is the antibody molecule. And that is why it is important in immunity, and it's also important in diagnostic and research also. A lot of antibodies, they are getting produced. In the research, you produce secondary antibodies, you produce primary antibodies, you do a lot of Western blotting, you do immunofluorescence, you do ELISA assays, where this is the antibody that is being produced and that is being used. So in research, I think this antibody is being used exponentially. right? So these are some of the important points regarding the IgG antibodies and its subtypes. I hope that you were able to understand all these points. And I'll bring another video, and where we will discuss similar topics, and in the same way quickly we will understand the main concept behind the topic, and we'll also understand the advanced information in a very, very easy and in a very, very illustration based video way. That is our own way. And please, if you like the video, then if you have watched this entire video, that means obviously you have liked the video, and then please do subscribe to the channel, and also hit the like button, share the video with others who can also get benefit from these kind of lectures. They are freely available on YouTube, so everyone should get benefit of these videos. And if you are interested in immunology and other related fields, I'm sure that this channel is going to be very, very useful for you if you're preparing for exams, if you're preparing for medical exams. I think all these videos are gonna fantastic for you to prepare for these exams. All right, so hopefully I'll also recover with the help of my immune system, including the IgG antibody, especially the IgG1. I'm looking forward to the function of IgG1, and hopefully I'll be recovered soon. Thank you so much for watching the video, and we'll meet in the next one. Take care, everyone.